Hi, everyone. Today we're going to talk about sleep. It's our second last lecture of the term. Finally, we're going to end next week talking about learning and memory before our final midterm. Today we'll talk about a number of topics with respect to sleep. We'll talk about how sleep is a number of aspects of our brain which act as clocks to guide our daily and yearly and hourly lives. We'll talk about the suprachiasmatic nucleus and how it seems to be a biological clock. Then we'll talk about sleep specifically and the various sleep stages and dreaming, and we'll go into ideas about what the purpose of sleep is, what the neural basis of sleep is, and some disorders of sleep. So a biorhythm is any inherent timing mechanism that controls or initiates our biological processes. And so it's something inside our brain that starts our day every day, for instance. It's linked to the cycle of both days and also seasons that are produced by the Earth rotating. And animals like us that live near the North or South poles are affected by seasonal changes. And so we and other animals have to have yearly cycles that we go through. For instance, the way that animals shed and grow their fur throughout the year. Plants also display rhythmic behavior. As you can see throughout the day, their leaves get bigger and smaller, so animals aren't unique in this aspect. As humans, we largely evolved near the equator where there is no change throughout the year. And so this is one explanation for why our biorhythms are dominated by circadian rhythms of daylight and night every day. So throughout the day, our pulse, our blood pressure, our body temperature, our cell division, and all these other aspects of our physiology wax and wane in their activity, as you know, by getting hungry rhythmically throughout the day. So as we know, the Earth is tilted a little bit, and this tilting creates these long summer nights and short winter nights for us up here in the North Hemisphere. Here's some examples of biological rhythms of different periods. So there's circadian rhythms, which happen every day, but there's also circannual rhythms, which are yearly, like the migration of birds. Then smaller than a day, there's ultradian, which are like our human eating cycles. And then infradian, which are more than a day, but less than a year, like female menstrual cycles. So circadian rhythm behavior isn't just driven by external cues, we know, because we can put people in rooms with no external cues and see that they still go through circadian rhythms. So there's something inside the organism that's controlling these rhythmic activities. There's a biological clock, some sort of system that times behavior and allows animals to anticipate events before they happen. So your body wakes up in time for the light to come out. And birds migrate before the weather gets cold. So humans' biological clock we've seen is synchronized to the day, and it makes predictions about what's going to happen tomorrow. So it lets us anticipate the start of the day, the end of the day, and the meal times, for instance. And it prepares our physiology and cognition to be attuned to those times. So we have a clock regulating our feeding and sleeping times, but also at a lower level, we have clocks that activate different gene expression in our cells throughout the day. A free running rhythm is the natural rhythm that our body has if you take away all the external cues. So without any external cues, the human body has its own rhythmic period that's actually a little longer than 24 hours. And so if you leave people in a bunker with no external lights or cues, you find that they end up going to sleep an hour later every night, and their sleep-wake cycle will shift an hour or so every day. But we know that this sleep cycle can also change depending on things like lighting. So here's an example of humans that are kept underground in a bunker. So their waking period begins to change without any cues. The purple line shows when they're awake. So they're awake, and here's the between 0 and 24, between 0 and 24, so it's showing 3 days. And so they progressively go to sleep a little later each night. And by the time 3 days are up, they end up waking up right when the people outside the bunker are going to bed. Hamsters are nocturnal, and to show that the external light can affect their free running rhythm, it's been shown that their periods get shorter than 24 hours in darkness and longer than 24 hours in brightness. Whereas diurnal, daytime dwelling sparrows have the opposite effect. So in their normal daytime light, if they have constant light, their free running period ends up being shorter than 24 hours. So these free running periods can also be influenced by external things like light. Here's an example of that sparrow in either constant darkness or constant light, showing that the free running rhythm can either be made longer than a day, leading the sparrow to wake up a little later each day, or shorter than a day, leading the sparrow to wake up a little earlier each day. So this brings us to the term Zeitgeber, or time giver in German. Zeitgeber is an external environmental event that can entrain or lock the timing of these biological rhythms, like light. So when we get up in the morning and we turn on our lights or open up our phone, it's resetting the clock each day. And the word entrainment refers to this idea of aligning the cycle. So it determines or modifies the period of the biorhythm. So a biological clock can be entrained to the daily light cycles, and this allows the animal to properly predict when the next morning is going to be. So entrainment of biological clocks by Zeitgebers explains how circadian rhythms can synchronize with things like changes in light throughout our year. So even though the time that the sun comes up changes throughout our year, our body can adapt. Actually, in the very extremes of our dark winters, they push the limits of our body's adaptability, as do the extreme light summer nights. Light pollution is an important aspect of these biological clocks. Our exposure as humans to artificial lighting now is disrupting our circadian rhythms. And this disruption of circadian rhythms can account for much of our deleterious behavior, like accidents and daytime fatigue and emotional state alterations, and as well as obesity, diabetes, and other disorders, because the change in your sleep can lead to changes in your metabolism. Jet lag is an important aspect of these biological rhythms that affects us as well. Jet lag is a disorientation or fatigue that results from rapidly traveling through time zones, so that you end up in a dark light cycle that's very different from the one that you were already entrained to by your existing Zeitgebers. Someone traveling from west to east has a more difficult time than someone going, say, from Toronto to Vancouver. Because when you leave Toronto and you end up in Vancouver, it's only a little later than when you first left Toronto. So you're just staying up a little bit later to go that direction. People that constantly are traveling in airplanes, like pilots, have persistent asynchronous rhythms, and these can be associated with stress and fatigue. And sports teams are also relevant here, because some teams have to travel quite far across many time zones from east to west to play, and then we'll have reduced. There's evidence that teams traveling from the west to the east coast are less likely to win. Here's a diagram from the textbook showing the difference here. Someone traveling from New York to Paris ends up arriving in Paris in the morning when they think it's nighttime, as opposed someone traveling from Paris to New York ends up arriving in New York just a little earlier than when they were originally going to go to bed, a little later than when they would have normally gone to bed. Okay, so that explains some aspects of our biological clocks. Next, we'll talk about some physiological clocks in the suprachiasmatic nucleus of our hypothalamus and how that affects our physiology. So the suprachiasmatic nucleus seems to be the pacemaker of our circadian rhythms. It's located just above the optic chiasm, which you recall is the crossing over of the optic nerves just under the orbital frontal cortex. We think of it as the master clock, and it entrains a bunch of other clocks that then control our movement and our eating, which we call slaves. This terminology of master and slave with respect to things like oscillators and computer hard drives is archaic. The intergeniculate leaflet of the suprachiasmatic nucleus, the intergeniculate nucleus and the pineal gland are two other areas of the brain that also display clock-like activity. But nearly every cell in the body has a clock. It changes its activity throughout the day. And thinking about which of these areas is the master clock that controls all the other is important. And so to understand if it's the master clock, we might try to remove it or damage it. So if we take the suprachiasmatic nucleus and we damage it, daily activities are quite haphazard. These cells increase metabolic activity during the light period of the day. They're also more electrically active during the light period. And if you take away all their input and output, they still maintain this rhythmicity. If you take them and put them in a petri dish, even they maintain this rhythmicity. And the textbook talks about an experiment in which they injected stem cells from the suprachiasmatic nucleus into an area of a rat's brain without any rhythmic cells, and they found that they were still rhythmic as well. If they're isolated, they each remain rhythmic on their own, but each cell's rhythmicity becomes different from one another. And so the timing has to be set so they can all synchronize together and both to the external Zeitgebers. So recall from our chapter on vision that there's a visual pathway in addition to the ones we studied called the retinohypothalamic tract. And this retinohypothalamic tract receives input from photosensitive retinal ganglion cells that are different from the other retinal ganglion cells in that they are responsive themselves, not the rods and cones, but them to blue light. And these retinal ganglion cells are even active in people with no rods and cones. They project their axons across the optic chiasm and then into the suprachiasmatic nucleus to an area called the core. And they synapse onto neurons, which then activate neurons in an area called the shell. And the suprachiasmatic nucleus is just adjacent to the third ventricle. The shell then sends its output to other parts of the brain and drives these so-called slave oscillators. It also receives input from other parts of the brain. So if we use our muscles a lot at a certain part of the day, we can entrain the rhythms in that way as well. Excuse me. The suprachiasmatic nucleus's rhythm is usually entrained by our morning and evening light, but it can also be entrained or disrupted by changes in lighting and things like arousal or moving or feeding. So getting up and having an early breakfast is a good way to start resetting your clock. There are other pathways which can influence this rhythm as well, like the intergeniculate leaflet and the raphe nucleus, which releases serotonin. And these are ways in which non-photic events, like things that aren't light-based, can also affect the timing of the suprachiasmatic nucleus. There's two separate groups of circadian neurons, it appears. There's M cells, which seem to be most active in the morning and need light for entrainment. And there's E cells, which seem to be more active in the evening and need darkness to entrain them. There's also individual differences in genes and gene activity, which can explain, for instance, morning people and night people. These are called chronotypes. And these can be due to differences in gene expression in these SCN shell neurons. Oh, here's the study about the transplantation of SCN neurons. So endogenous rhythms themselves are not learned. They're actually a physical property of the SCN neurons. They transplanted hamsters, they were. So they lesion their SCN and the hamsters then eat and sleep a normal amount, but their rhythmic nature disappears. So they don't have sleep-wake cycles. But then if you take the SCN cells from embryos and transplant them into these animals, that you get their circadian rhythms reestablished. So these external cells can reestablish that rhythm on their own. Now, what exactly is doing the ticking? What in the brain is causing the time? So there's some sort of feedback loop that's been proposed in a Nobel Prize winning set of research in which proteins are first made and then combined together, and then the combined protein called a dimer actually inhibits the original production of those components which make it. But then that inhibitory dimer degrades over time, and that degradation time leads to this rhythm. And so there's a textbook image about this showing the proteins being produced and then combining and then inhibiting their production and then degrading. So how can we think of this timing system? We can think the light entrains the suprachiasmatic nucleus. The suprachiasmatic nucleus then entrains a number of slave oscillators, each controlling their own behavior, one single behavior like body temperature, for instance. And the SCN can do this via hormones, proteins, or even neurotransmitters through direct axonal connections. We see light coming in, the photosensitive retinal ganglion cells pathway and training the suprachiasmatic nucleus so that it peaks at noon every day, say in this case. And then those suprachiasmatic nucleus cells and training oscillators throughout the brain that bias our motor activity, our eating, and our body temperature, amongst other things. So the SCN neurons send these connections to nuclei close by in the hypothalamus and the thalamus. And then these nuclei can pass on that entraining signal to other parts of the brain more broadly. It connects to the pituitary endocrine neurons, and that can control the release of hormones. And these can entrain our body tissues and organs. It also instructs the autonomic neurons in the spinal cord to inhibit the pineal gland from producing melatonin. And melatonin increases our sleepiness at night. The SCN cells themselves release hormones as well into the blood that can affect our sleep and wake cycles. So the SCN controls melatonin, we've seen, being released from the pineal gland. So the normal functioning is that during the dark phase of the circadian rhythm, melatonin is released by the pineal gland. This promotes sleep and influences their parasympathetic rest and digest system. The suprachiasmatic nucleus also controls the release of steroids like glucocorticoids from the adrenal glands, like adrenaline, so that they circulate during the light phase. And so it allows your pineal gland to release melatonin during the dark phase and your adrenal gland to produce adrenaline during the light phase. And this mobilizes glucose for cellular activity and makes us ready to fight or flight. Hamsters' production of melatonin affects their sex cycle by changing the production of hormones. So As the days lengthen in spring, these hamsters have a yearly rhythm in which they're more likely to mate during the springtime. And the gonads of the male hamsters grow and they release hormones that stimulate sexual behavior. So overall, in the spring, when their melatonin level is relatively low, their gonads enlarge and stimulate sexual behavior. But in the winter, as the days lengthen, melatonin increases and the gonads shrink, decreasing their interest in sex. So we can see that just from this change in daily amounts of melatonin, large scale physiological change can occur because of the hormone signaling. We've talked about chronotypes a little bit. Chronotypes are patterns of behavior that we see showing variation in circadian activities. For examples, some people are early risers and like getting up and getting everything done. Some people sleep in and like to stay up late and work. Call them larks and owls. And as we become able to do more genetic analysis, we can study the genes underlying these chronotypes, and we can understand why there's individual differences in these biological clocks and how they might relate to disease. So, for instance, prescribing people some amount of sleep might be different for people that are early risers or late sleepers. So, why would we want to modulate our cognitive activity and our emotional activity? Well, because these are metabolically high taxing activities, we want to activate them at optimal times during the day. If you only have a couple hours of highly focused attention to give throughout the day, you want to target that throughout the day when you'll be working. And so, for instance, one interesting aspect of this is that this is controlled by genetic switches. And so throughout the day, our genetic expression can change in the day and the night. So some of our cognitive activities can occur without influence by SCN rhythms, but it is generally adapted for our activity to occur at the right place and time. But as we get older, we end up with a poor ability to actively make our activities happen at the right times, and this impairs our daily schedule. And some things about our circadian rhythms could affect our emotional activity. For instance, our emotional responsiveness might change throughout the circadian rhythm as well. And this explains, for instance, why people might get scared at night. It might not just be from the darkness, but from their emotional rhythms going through a peak in susceptibility to fear at that time. Next, we'll go on and talk about the specific sleep stages and dreaming. There's variation in how long people sleep and their waking behavior. People sleep more when they're young. Most people sleep between 7 and 8 hours a night. And some sleep much, much less, sometimes as little as an hour. We can use a polygraph, which measures the electrical activity from the body over a number of sensors to measure the electrical activity from both the brain and body. This includes an EEG, an electroencephalogram, an electromyogram, a measure of muscle activity on the neck, and electrooculogram, a measure of eye movements around the eyes. Throughout the night, as we fall asleep, the frequency of our brain activity from the EEG changes. When we're awake, there's a high amplitude with a very fast frequency called a beta rhythm. That's about 20 times a second. and there's muscle activity and the eyes are moving around. As we get drowsy and relaxed, this changes to an alpha rhythm, which are larger amplitude. Sorry, the previous one was small amplitude. The next one, alpha rhythms, are larger amplitude. They're slower in frequency, around 10 times a second, but the muscles are still active, but the eyes are now closed. As we become drowsy, we switch to a theta rhythm where the waves increase and the frequencies get slower. The eyes aren't moving, but the muscles are still active. And then finally, a delta rhythm in the deepest stages of sleep. These are large, slow waves around 2 times a second, and they're associated with a complete loss of consciousness. And they still is muscle tone, but there's no eye movements at all. And these can all be contrasted with a type of sleep called REM sleep, or rapid eye movement sleep, in the textbook called R sleep. R sleep is a fast brainwave pattern displayed by the EEG, which involves complete muscle inactivity, except for some twitching eye movements. And much faster brainwave activity, more similar to a waking state. And this can be contrasted with this type of N sleep, where there's this large, slow frequency delta rhythm, and the eye movements are generally inactive. Body temperature usually changes as we sleep and it declines throughout the night. And as we've experienced, tossing and turning and laughing and dreaming all occur more often in specific sleep stages. So here's these 4 stages of non-REM sleep, 1, 2, 3, and 4, going from shallow to deep. And here's an example of that excited awake beta rhythm turning to an alpha rhythm, much slower and higher amplitude, and then decreasing in speed and increasing in amplitude even more to this deep sleep delta. If you have a Fitbit, you might get a graph like this every night showing your sleep stages. This shows over time which stage of sleep you were in. Here, waking, N1, N2, deep, N3, N2 up to REM sleep, back down to N3 to REM, N2 to REM, awake for a bit. And this shows a lot of stereotypical sleep behavior in humans. Generally, the beginning of our night is dominated by these deep sleep cycles, and the end of the night is dominated by rapid eye movement activity and much less deep sleep. And then we also see that these cycles occur between deep sleep and REM sleep, deep sleep and REM sleep, about every 90 minutes. And we've talked about all these things so far. Temperature's lowest during the earlier non-REM dominated part of the sleep, and then it goes up and matches closer to the normal body temperature during the REM dominated parts of sleep. People who sleep about 8 hours spend about 2 of those hours in REM sleep, and the duration of REM sleep varies with age, and it changes over a lifetime. It's very high, about half of the sleeping time in infancy, and it increases during growth spurts, after exercise, and during pregnancy. So here's showing a change with aging. The amount that we're awake changes drastically with aging, so that we end up only sleeping about 6 hours by the time we're 90. But also the proportion of REM sleep decreases as we get older as well. Non-REM sleep has a large range of associated activities, like a decrease in body temperature and an increase in growth hormone. Dreaming does occur in NEM sleep, but dreams are not as vivid as they are during REM sleep, and we generally don't wake up, so we don't remember them. But you can wake people up in the middle of these periods and find that they are dreaming. Periods of sleep talking, sleepwalking, and night terrors are all occurring during this non-REM sleep as well. People that talk or grind their teeth or flail around or kick their feet are doing so in non-REM sleep. That your muscle posture is generally maintained. And we know that we can sleep in a variety of postures, including sitting and even standing up. REM sleep, conversely, is associated with complete atonia, no muscle tone. So you completely lose your muscle tone and fall over if you fall into a REM sleep. Complete muscle activity is produced by the sleep regions of the brainstem inhibiting motor neurons. And the mechanisms that regulate body temperature also stop working, so the body temperature moves towards the room temperature. Dreaming is an important aspect of sleep. Vivid dreams occur during REM sleep. Everyone dreams a number of times each night. We can find out by waking them up during the dream. People often don't remember them after the fact. Dreams appear to take place in real time. And as the periods of REM increase, the dream sessions get longer and longer, therefore. And they know that it happens in real time because they've woken people up after a brief period of REM sleep and asked them how long it has been in their dream. The dreams during NEM sleep are generally less vivid, except for the ones that are associated with night terrors, sometimes in children. Night terrors are brief, very frightening dreams that can even persist after the child wakes. In terms of understanding what we are dreaming about, this is a much harder question to ask because it's kind of unfalsifiable. It's hard to make predictions and test them because most of the explanations are kind of post hoc. But one of the most classic interpretations of dreams is by Sigmund Freud and Carl Jung. Freud proposed that dreams are fulfillment of our unconscious wishes, and that they contain both the manifest content, this kind of loosely connected, bizarre stuff we see, but an underlying latent meaning. What that really means is that you are nervous about your class, for instance. Carl Jung was a contemporary of Freud and proposed that dreams were, instead of our own unconscious desires, were expressions of our collective unconsciousness, that the history of the human race was contained in our genetics, and that we could relive some of these meaningful historical lessons through these dreams. More objectively, we now know that dreams are related to quite recent events and ongoing problems. The color, symbols, and emotional content often relate to events taking place in a person's waking life. And there's two ideas about explaining dreams. One is a bottom-up idea that a person has a dream, and then the dreamer in some way analyzes the dream and interprets it, and we get meaning out this way. And alternatively, a top-down account would be more similar to Freud or Jung's, in that the dreamer actively constructs the dream. The bottom-up approach is more kind of random. A random amount of stuff gets presented to your brain, and then we make up a story to explain it. In terms of this idea of random activity, Hobson proposed this idea of activation synthesis. So the cortex just gets bombarded with signals which produce a waking EEG, and the cortex generates images and actions and emotions from memory associated with those activities. But because they're random, they're fragmented, and they're bizarre, and our brain has to kind of interpret them and synthesize them together into this idea. But oh yeah, that was a dream about this when we wake up. And so they're personal, this should say personal, and that our own memories and our own experiences are activated, but they don't have underlying inherent meaning we need to think more about. Alternatively, Antio Revenusio proposed that evolutionary idea that dreams are biased to help us deal with threats. and that they're useful because they lead to enhanced performance when we face those real threats in real life. So they're adaptive. But one idea is that we kind of experience all these problems that kind of carry over into our dreams, and then we attempt throughout the night to solve them through interpreting the dreams in this top-down approach. So the content of the dream can incorporate emotional problems of the dreamer, and these can lead to improvements or adjustments. When you are working on a difficult decision, you can be advised to sleep on it, and often the decision can be easier to make after. So this brings us to the idea of the function of sleep. What does sleep do? Sleep isn't a passive process. It's not just a turning off of the brain, and it doesn't occur just because of a decrease in sensory stimulation. And so if you isolate people and don't give them any stimulation, they'll end up sleeping actually less and less. And so we need our active experiences and excitement throughout the day and stress to necessitate sleep. One idea is that sleep conserves energy. So if we only gather food in the daylight, we might as well be offline during the darkness. And we can use that energy to better forage during the day and save it during the nighttime. So the higher nutrient diet an animal has, the less time they spend foraging and the more time they spend sleeping. Like a cat, for instance, is an extreme example. So predatory animals like cats sleep more than prey animals, and nocturnal and diurnal animals sleep when they can't easily travel. So humans sleep at night because we can't easily move around at night. So here showing various animals and their amount of sleep, showing the house cat sleeps almost the most, except for an opossum, which the proposes, even though they're, the opossum may have specifically evolved to conserve energy through this massive amount of sleeping. So as we know, sleep contributes to energy conservation because energy is not being expended while we're asleep. Also switching off the brain or decreasing the activity of the brain to these slow rhythms across the brain, not off, can conserve energy. Every cell is less active. And the decline in body temperature decreases the metabolism. One extreme of this is in hibernation in some animals. It's been proposed that there's something called a basic rest activity cycle in both our waking and sleeping life. These are recurring cycles, about 90 minutes, during which our arousal waxes and wanes. So there's lots of examples from our lives, like school classes, work periods, meal times, but also the cycles of non and REM sleep. So even at night, this cycle doesn't turn off. And we can see it here. So we sleep, and during our sleep, there's a number of cycles alternating between REM and non-REM sleep. And then we wake up, we go through these cycles of eating and resting about every 90 minutes. Sleep has been proposed to be restorative, and that there can be chemical events that provide energy to cells that get diminished throughout the day, and they need to be replenished during sleep. But alternatively, the feelings we have of fatigue and alertness may be kind of superfluous to that. They may just be aspects of our circadian rhythms, and they might not have to do with decrease in our physiological performance. In the past, some individuals have attempted to stay up as long as possible, up to 18 days, in which it's been shown there's been no physiological effect. I think the record in humans was 11 days. Sleep deprivation does lead to decreased cognitive performance the next day, though, and specifically tasks that require focused attention decline the more sleep deprivation we get. However, there's things called micro sleeps, which can confound this type of research, where people briefly sleep for a moment. We can deprive people of REM sleep by waking them up whenever they enter it to find out what its role is. One finding is that there's an increased tendency to enter REM sleep when you take it away, and it becomes harder and harder to deprive people of REM sleep. And the next night, there's going to be more REM sleep when you stop doing this, so there's a rebound. But there's no adverse effects of REM sleep deprivation. Many antidepressants reduce REM sleep, but there's no evidence of adverse consequences. And also some brainstem damage can completely remove REM sleep without any apparent problems. And it might be that the ways we're measuring things aren't sensitive enough to these changes, and that the sleep deprivation might affect people at some distant time in the future. Another theory is that sleep helps us to solidify our memory, and we'll go on and talk about these ideas next week in our chapter on learning and memory as well. But the idea is that memory storage requires 3 phases. There's a fragile, labile phase where it competes with other memories and the other new memories we make. There's a consolidation phase where a relatively permanent representation is made, and chemical and genetic activity must lead to structural changes in the brain to lead to this permanent change. And then there's a recall phase where the memory is used at some time in the future. And this can also further integrate it into memory. So one idea of sleep is that there's different kinds of these memories are stored during different sleep states, or that memory is manipulated in different ways during the various sleep states to allow for this consolidation. One evidence for this is that the brain regions that are associated with creating a memory while someone is awake are also active while someone is asleep. Some more evidence that sleep and explicit memory are linked comes from place cells. These are hippocampal neurons that fire whenever a rat is in one location of their environment. So they're in a big box, and whenever they go to one corner of the box, this cell will fire. These have been proposed to be related to kind of a GPS map in our brain. We can record from about 100 different cells at the same time during non-REM sleep, during food search, and during non-REM sleep after the food search. And after the food search, their activity, the correlation between those 100 neurons was very similar to how it looked during the food search. So here they're searching for food, and some of these neurons are highly correlated. Those same highly correlated neurons are active during non-REM sleep the next night, whereas they weren't active the night before during non-REM sleep. Indicating, for instance, that there might be some replaying of the memory during sleep to further consolidate it into memory. Marquette used PET scanning to record brain activity while humans were performing a reaction time task in which there was secretly an unconscious order in which people were learning, and they got faster and faster without knowing it. And the imaging during the sleep after this learning revealed that regions that were active when they were learning the task were also active during their REM sleep. And the authors proposed this meant that the subjects were dreaming about what they had learned, and that this dreaming strengthened the memory. Here's a diagram showing that increased blood flow during the reaction time task, and then in the next night as well. So we can strengthen memories during sleep, but we don't know if the same molecular and neural changes that support memory storage during sleep resemble those same ones that occur during wakefulness. Okay, next we'll talk about the neural basis of sleep. Melatonin is a chemical that's secreted by the pineal gland during the dark phase, which causes sleepiness in us. And a synthetic form can be taken as a sleep aid. So it can be thought of as a sleep-producing substance. The reticular activating system, we've learned in our anatomy section, is this large structure in the middle of the brainstem, a mixture of nuclei and fibers. It's associated with sleep-wake behavior and arousal. It's called the reticular formation as well. If you stimulate it, you produce a waking EEG, and if you damage it, you can produce slow waves similar to sleeping, and this can lead to a coma. And so many times when brain injury leads to lasting comas, it's from damage to this particular activating system. If you cut the incoming sensory pathways to the reticular activating system, it still produces the desynchronization of the EEG. And so the incoming sensory stimuli activate the cortex through this reticular activating system. Two other brainstem systems influence our waking life. The basal forebrain contains acetylcholine cells that secrete acetylcholine onto cortical neurons, and these stimulate waking EEG. And at the same time, the median raphe nuclei in the midbrain that release serotonin project it to the neocortex, and these also stimulate beta rhythms. So two areas of the brainstem are sending projections to the brain to wake it up. So the basal forebrain neurons produce activated EEG pattern when the rat is alert, but when they're not moving. And the raphe neurons produce active EEG patterns when the rat moves. And this kind of brings us to this idea when you're on a long car ride and you're starting to fall asleep, if you start to move around, you can use this second system and release serotonin into your brain as well and increase your wakefulness. If you damage either of these areas, you lead to EEG that resembles a sleeping animal, especially if both of them are destroyed. And if they're both destroyed, the animal can still walk around, but it can't learn or display intelligent behavior. So the reticular activating system seems to produce its arousing effects by influencing these two pathways. Additional two areas that are related to sleep are the prebrachial area. These are cholinergic nucleus in the dorsal brainstem, which seems to be related to our sleep. It projects to the medial pontine reticulum and seems to initiate the REM sleep and other REM-related behaviors like the atonia. The medial pontine reticular formation is a nucleus in the pons. It also participates in our sleep. It projects to other brain regions that produce these REM-related behaviors like the atonia during sleep. So these regions come on driven by the suprachiasmatic nucleus at the night and induce us to sleep and induce this REM sleep every 90 minutes. Damage to either of these areas reduces or abolishes REM sleep. So the prebrachial area seems to initiate sleep. The medial pontine reticular formation can produce REM-related activities like activating EEG via the basal forebrain nuclei and exciting the brainstem nuclei to produce REM and other twitching movements. This also leads to a loss of muscle tone produced by the subcurricular nucleus, which excites the magnocellular nucleus of the medulla and inhibits the spinal motor neurons going down to the body. Finally, we're going to end with disorders of sleep. Insomnia is a disorder of slow wave sleep where you have a prolonged inability to fall asleep. It has multiple causes. There's also drug dependence insomnia, where we take a drug like a sleeping pill and we need more and more of the drug to get the same benefit. And the tolerance also results in deprivation of certain stages of sleep, and this leads the user to increase their drug dosage in a vicious cycle. Narcolepsy is a slow wave sleep disorder in which a person can suddenly fall asleep at inappropriate times, especially exciting times. Drugs that stimulate dopamine, like Ritalin, seem to help with narcolepsy. Sleep apnea is a very common sleep disorder in which people are unable to breathe due to either obstruction of their airway or due to central pattern generators in their brainstem. And the person has to wake up to breathe every couple minutes, and it can lead to terrible sleeps. Sleep paralysis is a disorder in which the atonia and dreaming of REM sleep are occurring, but a person is awake. It usually occurs when people are just falling asleep or just waking up. Cataplexy is a form of narcolepsy in which strong emotional stimulation leads to the narcolepsy. And the animal loses all their muscle tone as if they're in REM sleep instead of in deep sleep. A hypnagogic hallucination occurs just before you fall asleep or just when you're waking up, and it's a dream-like event while a person is in this state of cataplexy. And these can obviously be related to hallucinations that we see from the ingestion of different drugs, which are probably have a similar physiological origin. Orexin cells in the hypothalamus send these projections to other brain regions, as do the other areas we saw in the acetylcholine and serotonin projections. Orexin seems to play a role in maintaining waking as well. And so if you have a loss of orexin, it can be related to cataplexy. Some people have a disorder called R-sleep behavioral disorder who behave as if they're acting out their dreams. This can affect people who take antidepressant drugs, and it can be treated with benzodiazepines, an anti-anxiety drug that blocks REM sleep. So while they're in REM sleep, their body isn't frozen and they'll actually act out. Finally, we want to think about what sleep tells us about consciousness. Every day we go through these stages of sleep that are a great indication of that our consciousness can change. And so studying the difference in brain activity from being awake to being asleep and dreaming can help us understand what type of physiological activity gives rise to consciousness. Consciousness isn't a unitary phenomenon. There's different states of consciousness one can be in. Studying sleep can also help us understand psychiatric and drug conditions. For instance, like I just mentioned, are the hallucinations that we see during dreams similar to those that we see in, for instance, mental disorders? I forgot to put this slide at the end, so I'll just finish on this. So today we talked about the clocks of our body and how they relate to the changes in the outside world. We talked about the suprachiasmatic nucleus and how it can control other oscillators in the brain. Talked about sleep stages and dreaming, and then the function of sleep, the neural basis of sleep, and some disorders. Thanks very much.